What's it like working with coronavirus and other deadly biohazards? The future in the fight against COVID-19, a glimmer of hope, and using light to control genetic expression. Join me in this episode for a thrilling look into the life of a scientist on the front lines during this pandemic, as well as for advice for young scientists and professionals alike. This is Goggles Off. What's up, everybody? Welcome to another episode of Goggles Off. Today I'm joined by Dr. Eric Valois. Eric, how are you doing?
I'm doing great, Brandon. How are you?
Fantastic. Eric, you received your PhD in biochemistry and molecular biology from the University of California, Santa Barbara. Can you speak to that a little bit? What did you study during your doctorate studies?
So I was advised by Dr. Herb Waite, who is a forefront expert in biomimetic materials and marine adhesion. So most of my work focused on looking at the muscle byssus, and the byssus is that little thing that you've probably seen if you've been to the beach. It's what anchors the muscle to the actual rock. It looks like a little thread. So I studied, how those threads stay intact, and I studied how those things actually adhere to the rocks themselves.
Gotcha. Very cool. We're not actually going to talk about that a lot today, but I did want to, since it is really cool research in itself, I wanted to kind of show that and highlight the diversity of science and, what a scientist does. But today, our subject of conversation is a little bit more relevant to today's times, which is the coronavirus and your recent work to try to develop a orthogonal or new method of testing, improving upon the current standard used method of testing. Coronavirus represents an unprecedented health crisis. 2 million cases in the United States, roughly 110,000 deaths. Because of this, the government is putting a whole bunch of, social distancing orders in place. And it's really hard to say with certainty how effective this social distancing and these measures are. They, for one, cause a huge social and economic strain on the economy, and we really don't know how well they're working. And that's partly because widespread testing is so difficult to achieve, and it's a logistical problem. I know for a fact that when this all kind of broke out, I came down with the sickness, and I had all the symptoms of coronavirus. And I remember calling the hospital and inquiring, hey, I want to get a test, this and that. And it's Kaiser Permanente, so huge hospital, like very, very well renowned. However, they told me they don't have any tests and they can't do anything for me. And I just kind of had to ride it out, especially because I'm not one of these at-risk characters. And so making a new test or improving upon the current testing procedures is critical. And I was wondering if you could describe this process a little bit.
Yeah, absolutely. So I think that you're absolutely right that widespread testing was a huge systemic problem early on during the coronavirus. The explosion of cases that we saw back in February and March was astronomical, and it hit the supply chains extremely hard and kind of depleted all the resources that we had. So the current gold standard put forward by the CDC is a technique known as RT-qPCR, which stands for real-time quantitative polymerase chain reaction, which is kind of how you convert RNA into DNA and then quantify how much of that is there. So you can really figure out how much virus a person has kind of circulating in the back of their throat. So the really big issue there is that process was developed to be one step, where it's this solution that you can buy off the shelf and run all these tests. The problem was the CDC put forward a very limited number of suppliers that they approved. And so all the hospitals went to the same suppliers to buy this mix of reagents. And so, these suppliers sold out extremely quickly. And then there was a backlog for months. And so the CDC approved tests really were limited in number. So they were reserved for the people, A, most at risk, and B, that were actually presenting symptoms of really severe cases. So our goal here at UCSB, and this was a collaborative effort between Dr. Ken Kosick, Dr. Max Wilson, Dr. Diego Acosta-Alavar, and Dr. Carolina Arias. We met and we decided that our goal was to create a test that could circumvent the classic supply chains. So in doing so, it opened up testing to, much broader populations because you didn't rely on a single supplier to give you reagents. Our test is very similar to the CDC's base test, which is called the TACMAN. Our test is an RT-PCR method followed by fluorescence readout that is generated by a CRISPR enzyme. And, many people have heard about CRISPR. It's the genetic or genome editing protein that allows us to do a lot of really interesting things. So the fundamental way that our test works is we harvest RNA from, the patients. We take that RNA, we reverse transcribe it, which is the RT part of the qPCR. We reverse transcribe it from RNA into DNA. We amplify that DNA, and then we feed it to this Cas13 molecule, which will then transcribe it again back into RNA, cleave that RNA, and you get a fluorescent signal. So our readout is fluorescence. So really all you need to get, a result being positive or negative is a blue LED, because if you, shine a blue LED on this tube, it'll either fluoresce if you're positive or it won't fluoresce if you're negative. So it's kind of the fundamentals of our test and what we went to go about.
Very cool. And kind of platforming off what you said, really the ways your test is superior or makes improvements on the current issues regarding the gold standard that's, of RT-qPCR, would be it addresses, 1, the availability of the resources, like you said, 2, the availability of equipment, because the equipment used in RT-qPCR often really expensive, like a commercial thermocycler. I looked it up on Biorad and could find one for like $5,000, and then a spectrofluorimeter is
$30 plus.
$30,000, yeah. So I mean that's a lot of money. However, the stuff that you guys were using in the lab, this little mini thermocycler that I found right there on Thermo Fisher for $500, and then also that fluorimeter you're talking about is this little actual cardboard box that costs $28. So roughly, more than a 10-fold decrease in the price, and that opens up, different niches. Now maybe smaller labs can actually start doing testing, or, even do-it-yourself scientists at home can even start doing testing. So really broadens the amount of people that can administer these tests.
Yeah, I think it's been really phenomenal to see kind of how the industry has responded to demands in reagents. No free ad reads, but here we go. New England Biolabs is where we're buying all of our things, most of our reagents, I should say. And New England Biolabs is a massive company out in Massachusetts. But we're ordering off-the-shelf items. We're not ordering anything customizable. We didn't sign any, contracts. We didn't negotiate any novel prices. It's just what they had available, which means that anyone else has the same resources that we had available. And they ship usually in, 2 to 3 days. So all of the resources that we need are coming rapidly, which is phenomenal, and it's a great comparison to what we were experiencing early on in coronavirus testing. And like you mentioned, the cardboard box that we use was designed by a company called Mini PCR, and Mini PCR is I believe they were founded by a bunch of MIT grads, but their whole goal was to bring science to elementary and middle school classrooms. So they developed this cardboard box that would generate fluorescent signals. They generated cheap thermocyclers that you mentioned, were about $500 to $700 depending on the model that you bought. But again, they're able to generate large scale, cheap, effective equipment that could be rolled out into any classroom. So we figured that this would be a great way to kind of utilize equipment that is, like you mentioned, orthogonal to the standard methods.
Yeah, very cool. And you mentioned it's being effective, and it really is just as effective as the current gold standard set out by the CDC, which is the RT-qPCR, like we talked about, and achieving it with just a much lower cost and an ease of use. And then also touching back to what you said, this could be something that all you really need is an LED, blue light to light up, and it gives you your positive result. And so this really represents a point of care solution to medical diagnostics, right? So as opposed to the current method where you go into the hospital, the doctor sees that you're sick, orders a few tests, then you wait for the lab to get those test results back to you. Usually takes like 6 days or so. Point of care is really this idea that you go to the doctor, and right there at the point of care where the doctor is seeing you, they do some sort of quick little test right there, get the result right there, and can tell you, do you have coronavirus or do you have this infection or not. And with regards to medical diagnosis, right, time of diagnosis is so key, right, because then that can inform the decisions of public health officials and also of an individual themselves as to how they should go about themselves in their day. So it just is really interesting, kind of an advancement in the medical diagnostic field.
It is, and I think that's the ultimate goal, is getting to that point of care testing. With that being said, I think the little caveat that we have right now is our fluorescence readouts are really only about 8 tests. In that little cardboard box. We just would need to do a little bit of engineering to make a bigger box, but that's kind of a trivial task to any, mechanical engineer. This test that we're doing now, the CREST method, as we call it, has been scaled pretty largely, and we're running about 150 tests to 200 tests a day. So as you mentioned, point of care is pretty important to us, and at 200 tests a day, we're getting results in 24 hours. So this is something that's easily handleable by a university medical center for student testing, or small hospitals in rural areas. It's definitely something that can help impact point of care in smaller urban areas.
And then you mentioned CREST, just to break down that acronym for people. That stands for Cas13 based, rugged, equitable, scalable testing. So kind of exactly what it sounds like. The technology is based off the Cas13 proteins, like we talked about earlier. It's rugged, meaning a lot of people can do it, and it's able to be done in a diverse array of conditions. Equitable in results to the current gold standard, RT-qPCR, and scalable, meaning that you can scale it up as you wish.
I think the scalability is the big one here. While all the letters of the acronym are important, the kind of crux of any diagnostic testing is your ability to generate a signal, right? A positive or a negative signal. In our case, the signal is generated by this Cas13 protein, which is that CRISPR protein, and we've been able to generate hundreds of thousands of tests worth of Cas13 in about 4 days. So we can generate huge amounts of signal producing Cas13, which is also really cool. And just getting back to the rugged part of things, the thermocyclers we use are actually capable of being battery operated. Oh. So in theory, you can run this anywhere. You don't need your standard power supplies, which is kind of cool.
That is really cool. Kind of like field laboratory diagnostics right there in your pocket. That's really cool.
Yeah, it's actually getting to that point, the field deployability. We've actually sent 200 tests to Ecuador, which are being used to test native populations in the jungle down there.
That's really cool.
So all these people are using battery-operated detection mechanisms and battery-operated thermocyclers, which is kind of fun. So we're awaiting those results. We haven't seen them yet, but I'm hoping that we can get this, and maybe we can update listeners at some other point.
That's very cool. It kind of reminds me of something I was researching in undergrad, but it was during the West Nile Ebola outbreak. Researchers were trying to pin down where the Ebola virus was coming from and originating from, and there's differences in the virus's genetics based off where it originated from, and so they unleashed these brand new kind of little pocket DNA sequencer machines that they would actually go out in the field and take a swab of someone's DNA, plug that little pocket DNA sequencer into a computer, and get a result, and then be able to actually pinpoint what regions of Africa they actually had to shut down at that time. Just really cool, this idea of field deployable scientists. It makes me kind of feel like less of a science nerd in the lab, and maybe someday I'll be like an action hero, out there deployed in the field.
I mean, it is kind of cool, right? Like how far science has really come. And kind of getting back on topic to the coronavirus, we've actually, not we personally, our lab, but the science community as a we, has actually been able to do that with this coronavirus, where they've been able to trace genetic lineages from continent to continent. So we know, for instance,
that the genetic strains of the coronavirus that hit Washington and California originated in Asia, where the genetic strains of coronavirus that hit New York originated in Europe. So we could actually track, how the virus spread from east to west and then west to east. So kind of an interesting thing. It's kind of amazing. And like you said, field deployability would be, I think that's every scientist's dream, is getting out there and doing something that's hands-on, muggy, and kind of gross at times, but I think fun in the end. It's nobody, I shouldn't say nobody, but being in a lab nonstop all the time can be grueling. So it's always kind of fun to think and dream that your science can be applicable on the ground.
Yeah, it's very cool stuff. What is it like working with such a pathogenic virus on a day-to-day basis? What kind of precautions did you have to take? Was it ever kind of scary?
Yeah, I think I'd be lying if I said that there wasn't some innate or intrinsic fear. I think working with pathogenic materials, if you're not somewhat afraid of what you're working with, then you're bound to make mistakes. If you're nonchalant and careless, that's when people get hurt. With that being said, the team that I'm working with has put in great standard operating procedures to make sure that there are absolutely minimal risk of exposure to a live virus. So I can kind of walk you through that if you want. I can tell you about that.
I'm super interested. How does one prevent themselves from getting sick from the thing that they're studying at the lab bench?
Yeah, so the big one is utilizing PPE, and that acronym, personal protective equipment, has been all over the news. We hear about it all the time, especially with the shortages. But for us, the PPE is an N95 respirator, full face goggles, so not a face shield, but actually sealable goggles, a barrier lab coat, which is impenetrable to fluids, and then we double glove with nitrile gloves. All of our work is done in a device called a biosafety cabinet. Biosafety cabinets are different from your classic chemistry hood that you see in undergrad biology and stuff like that, in the sense that the flow is all into the hood, and then it's filtered through two HEPA filters, which are designed to capture any biological material. So the biggest risk of infection is actually transporting the samples from sample collection site to the lab. Once they're in the lab, they're immediately deactivated. We essentially bake them, which will rupture the virus capsid and render them inactive. But like I said, it's transporting them from collection site to processing site. With that being said, the samples are minimally touched once they're collected. They're placed in individual tubes. Those tubes go into a rack, which is sealed in a essentially a biological transport cooler, and it's not reopened until it is actually in the hood itself. So, like I said, there's always a little bit of fear. You do your best to make sure that you have followed your PPE and standard operating procedures, but I don't know. We've nailed it down pretty well. So the other thing too, I guess the really important thing here is we always are working in 2-man teams. So that way, the cooler comes in to our biosafety level 2 facility. It goes into the hood. There's one person that's actually handling the samples. Their arms never leave the hood. The second person is reading and recording numbers. And so let's say that I'm working in the hood at the time, I'll pull a tube out and it'll say sample 1111, right? I'll read off that sample. My partner will repeat it. It'll get logged, and then it immediately gets activated. So if there were any sort of problem, there's always 2 people there to deal with the issue. So that way we never lose track of samples. We never have problems. It makes handling samples like that much more comfortable to know that you're A, not working alone, and B, you have someone there to kind of reinforce what you're doing.
Has there been any issues? Has anybody from your lab gotten sick during the course of this research?
No, actually. No one's gotten sick, which has been great. We had one random case of bronchitis, but yeah, tested twice for coronavirus, both times negative, which is kind of crazy. But they, for some reason ended up with bronchitis, which is just like horrible luck, I guess, to get a respiratory infection in the time of coronavirus. It's not coronavirus. But no one has actually contracted coronavirus. Like I said, we have been immensely careful.
That's awesome. And I didn't even think about it, but it makes sense to have one person scribing down everything that you're saying and logging it, because there's no way you could be efficiently cleaning and scrubbing your gloves from working with the virus and then go log into your notebook without kind of contaminating something.
Yeah, I think the other thing that's really important about that is like once the flow hood or the flow of air in these hoods is very delicate. You're about 100 cubic feet a minute, which sounds like a lot, but it's not, especially when your hood is 6 feet wide. So once your hands go into the hood, you want to minimize the number of times that they come out. So, you'll be sitting in the hood for 2 hours with your hands in the hood the entire time. And so you want to make sure that person that's your support is, helping. Like if you need a new set of pipette tips, like they grab them, you don't exit the hood. If you need, ethanol to like clean your gloves, they'll supply it for you. If they need new buffers or new reagents, they'll be the one to grab them. So that way you don't disrupt the flow of oxygen and you minimize the number of times that you break that airflow. So it helps also not just like reiterate what you're doing, but it helps actually improve the safety of your operation.
Does it get tiring to hold your hands like that in the hood for an extended period of time?
Yeah, it can. 2 hours is fine. The longest day that we had was about 9.5 hours in the hood. And that was pretty grueling, but all in all, we've all done this. Anyone who does like cell culture can tell you it's not the most exciting thing to be in the hood, but it's not overly tiresome. I think if I had to do it alone, that would be brutal.
And probably very unsafe from the way you're describing it.
Yeah, exactly. But it's not too bad. Like, it is what it is.
Okay, well, and how do you feel about, traditionally, you did the muscle foot protein stuff, and then in Max Wilson's lab, we haven't talked about it yet, but you did a lot of kind of optoprotein investigations. But kind of, are you enjoying this excited phase and, and working kind of pathogens, or would you like to get back to kind of more relaxed work?
It's been exciting. It's been an exciting opportunity. In Dr. Waite's group, I didn't do a whole lot of molecular biology. And when I talk about molecular biology, I mean really handling genetic material, RNA, DNA. So for me, this has been not only an opportunity to give back to the community that got me through my PhD, but it's been an opportunity to learn a lot about, molecular bio, from, cloning Cas13 to harvesting RNA DNA to doing RT-qPCR. Like, it's been a really awesome opportunity, and I'm just happy to have a job at the time. But yeah, I am looking forward to the time that I can return to work that is a little bit more up my alley. But I don't know. I'm happy to do what I'm doing for now. So we'll chug along until we can make significant progress and hopefully help our community return to some sort of normalcy.
Very cool. And then kind of touching off that, how do you feel about this idea of being a scientist and kind of, getting a diverse project every once in a while, or at a switch in your life, or the ability to kind of branch out and do multiple things at once? What does that mean to you?
Yeah, for me, I think it's ultra important. I value learning new techniques and new skills. The one caveat to that is there's always a steep learning curve. Education is kind of a pyramid, if you want to take a look at it, where you start at the bottom and you kind of cast this wide net of knowledge. Where you're in elementary school, you're learning everything. You're learning how to read, how to write, what numbers are, and basic geography. As you kind of progress through your education, that focus narrows. High school, you kind of get a little bit more in depth, so your pyramid narrows. You get to college, you pick a major, it narrows even further. And then you get to your PhD and you're at the top of the pyramid where you know a whole lot about one very specific thing. And for me, I think a lot of people kind of fall into that trap where they never to continue to expand their knowledge base. So for me, I value kind of learning new things from time to time. But like I said, it's a steep learning curve because you've got to step outside your comfort zone and like really force yourself to learn a new material or new topic.
Right. A lot of drinking coffee and reading articles that you don't really understand.
Tons of coffee, man. Oh my gosh. You would not believe.
And kind of platforming off that as well, what's it like being a postdoc? Now that you've completed your PhD, now you're doing a postdoctorate in Max Wilson's lab. Has that been very similar to the PhD in terms of work input, or is it just totally different? What's that like? It's
totally different. It's so much more work.
Really?
Yeah. I had somebody a long time ago kind of tell me that like each step you take in your education career, you not only increase your knowledge, but you increase your work ethic, or your workload, I should say. Hopefully your work ethic to go with it. But it's totally true. As a graduate student, you rely really heavily on your advisor to help you with grant writing, with paper writing, with things like that. As a postdoc, it's a really interesting transition because one day you're a grad student, the next day you're a postdoc, you have gained no additional experience, but the demands of you increase exponentially. You're now expected to write your own grants. You're now expected to like do your own budgeting. You're expected to develop multiple projects, not just a single project like you do during your PhD thesis. So it's been a lot of work, but at the same time, I think it's a really fun challenge. Like, I don't want to discourage any PhD student from giving it a shot, because obviously if you went through a PhD, you can get through this. That initial couple of months is just a kind of a shock to your system. Like, you close your PhD and you work immensely on getting your thesis done, because if my experience is typical, you put off your dissertation writing until like, I don't know, 4 months before your graduation date, and your committee's sending you emails like, hey, where's the draft? And you're like, oh, I'll have it to you tomorrow, and you're up till 3 a.m. writing. But you expect a little bit of downtime. If you jump straight into a postdoc, there's just no downtime.
And that is typical. Everybody I've interviewed has written their thesis in a very short period of time. So
yeah.
It's not uncommon to do that.
I don't know. Hopefully for all you young PhD listeners out there, start writing early. Like, get it done ASAP. Draft your papers in a dissertation form. That way all you have to do at the end of your work is just copy and paste your papers in your dissertation. You don't have to stress about writing it and applying for fellowships and applying for jobs, and you want to be able to enjoy the last few months of your PhD.
Definitely. Yeah, great advice. You got any for kind of a young scientist, someone maybe just starting off, maybe as an undergraduate or maybe a high schooler looking to get into science?
Yeah, I got advice for everybody. I don't know if it's good advice, but it's advice that I wish that I would have been given. I think for especially young scientists, and we're talking kind of freshmen here, freshmen, sophomores, is get it involved early, like as early as you can. Most people, like graduate students, don't want to take on an undergraduate that they are only going to get for 3 quarters. They want to be able to train you and have you be productive for a couple years. The easiest way to get a lab position as an undergrad is be available your freshman year summer. So you come in, you have no classes between your freshman and sophomore year. You get trained every day for 3 months. So that way your sophomore year, you can hit the ground running. You can manage a project, the techniques, where all the chemicals are in the lab, who to talk to, and that way you can actually be a productive member of your lab rather than being a burden to the grad student that you're supposed to be helping. So that's kind of advice for, getting into science. The other advice for graduate or undergrads looking for labs is when you contact these people, make sure that you have done your research. Like, don't send them a generic email that says, hi, my name is Brandon Mulady and I really like stem cells. and you're going to get an email back from a chemical engineering professor that's like, yeah, we do chemical synthesis, no stem cells, but like, thanks. Make sure you tailor each letter to each lab that you're applying to. Do your homework, because it'll go a long way. I don't know, you want me to go to grad students? I can do that.
I kind of wanted to touch on that.
Yeah, go.
There's been a bunch of undergrads who have kind of approached me because I was in a research lab and I had kind of been working, in a lab for some time now. And they had asked me, oh, Brandon, how do I get in the lab? And they would send me those generic emails. They're like, hey, I'm thinking of sending this to a professor. And
Dude,
it's like,
they're brutal.
It's like, dude, you just, or, guy or girl, but it's like, you didn't put in any thought into, their research. You know how many people are doing the same thing every day? Like, you need to read all their work, or at least, a good chunk of it, so you're knowledgeable in what they research. And so you can actually engage in a dialogue with this person about the research. And you should probably ask some questions that show competence and a thorough understanding of the material, and that you are seeking to advance the material, so don't just ever come at them like, oh yeah, like you said, like, oh, I like stem cells, because they're going to tell you to kick rocks. Like, everybody is interested in that right now, and so they don't have space for you. But if you come at them with some insightful questions that really show that you're a go-getter and kind of got a good head on your shoulders, then they're much more likely to reach out to you. So
Absolutely.
But yeah, graduate students, you got any advice for graduate students?
Yeah, grad students, first and second year grad students, you don't know everything. Like,
I see a lot of grad students and rotation students, especially, who like come into a lab and feel like they're going to cure cancer. And it's like, there's a reason that these really hard questions haven't been answered yet. It's because they're hard. Like, you don't know what you're doing when you're a first year. You fumble around, and granted, there's always exceptions. There's always people that are phenomenal, but I would say 90% of first and second years fumble. They take on tasks that are way too large for their talents or skill sets. And then they get bogged down, they struggle, they get discouraged, and that's just kind of this bad cycle. As a first and second year, you need to do two things. You need to pair yourself with an advanced grad student or postdoc and just get knowledge, like learn techniques, learn data interpretation, learn how to manage experiments, design appropriate controls, etc. And then two, you need to work on side projects that are just seem stupidly easy. Like, you need to have a win every day, whether that's just like have a gel that looks phenomenal, or do a Western blot that works, or like run a qPCR that shows you curves that you expected. Like, if you can get those wins and those successes, no matter how small, early, they'll build into something eventually. They will build into a nice story or culminate in a nice small paper. And like, if you can get one of those out in your first or second year, that's great. Like, you don't need to hit the home run that's Cell Science or Nature. Like, you just need to get something small accomplished early to set your foundation. And I think I was guilty of that for sure. Like, I came in to Herb's group and was like, I have this brilliant idea of solving how the muscle byssus attaches to the muscle plaque, or the byssus thread attaches to the plaque. And Herb was like, yeah, that sounds phenomenal. No one's been able to do it. I was like, perfect, I'm going to do it. Herb's been working on this problem since he was at Duke back in the 70s. If like Herb can't figure it out, believe me, there's nothing that I'm going to do as a first year that's going to do it. And I don't know, it's just, it never worked out for me because I was too ambitious. Like, I bit off more than I can chew, and I didn't have the skill set necessary. And I think a lot of first year grad students suffer from that problem.
I'll definitely try to heed that advice, because I'm going to apply to graduate programs pretty soon, so that section was a lot for me. I kind of want to touch on, how did you first get into science? Were you always scientifically inclined? Is there something that kind of triggered that?
I don't know if there was one like defining event, to be honest with you. I always found science interesting. To be honest with you, I was more math oriented in high school. Was not great at biology in high school, and then I got to college and really thought I was going the med school route. Like, that was kind of what I had geared myself towards, hence the kind of undergrad degree that I pursued. but I found myself being more and more attracted to the science, the experiments in biolabs and chem labs and things like that. And when it came time to write essays for the MCAT, I had a really big problem. Like, I couldn't explain why I wanted to go to med school, other than I thought it was like, I don't know, medicine sounds interesting kind of thing. And so that was really kind of eye-opening for me as a junior in college, was like, if you can't explain why you want to do something, then you probably shouldn't be doing it.
Right, for the rest of your life.
Yeah, exactly. So anyways, that was kind of, I found out that like medicine wasn't for me. And so I started thinking about what I really wanted to do, and science was always intriguing and interesting. Like, the ability to work on cutting edge things that changed rapidly, I found really cool. Like, I wanted to be on the forefront of whatever was coming. On top of that, to make a long story long, when I was going through undergrad, Obama entered his first term, and they dumped a bunch of money into the Brain Initiative, where they wanted to map the, all neuronal connections in the human brain, which sounded crazy, but they dumped a ton of money into it. And UC Davis founded the Mind Institute, which was like this huge institute attached to the UC Davis Medical School that like opened up a ton of neuroscience research. And so I got really involved in neuroscience research and loved it. And that kind of just like sparked my, I don't know, furthering interest in science, and like kind of the dedication that I wanted to spend to doing it as a lifelong career.
Very cool. So generally just find it interesting and cool. What else are you into? You got any hobbies, anything you like to do?
Yeah, sports. Big sports guy.
What sports are you
big
into?
Sports guy. I play ice hockey, which is very uncommon growing up in California. Play a ton of ice hockey. I love lacrosse, and then I surf, obviously.
How does one even get into ice hockey in California? It seems...
Yeah. It seems not common, and it isn't. When I was 9, I went to a birthday party at an ice rink and loved skating. And that kind of just blossomed into this, I don't know, just like obsession with skating. So, yeah, played all over the States, played in Canada. Just loved it. Still skate 3 or 4 times a week. Beats going to the gym any day.
And then you also mentioned lacrosse. What about, I mean, you were U.S. national champ in lacrosse. What is that? Was that in high school?
No, that was in college.
Oh, really?
Yeah, and that was as a coach, actually. So it was kind of interesting. Yeah, I guess I should talk about how I got there.
That was a weird scenario. Hockey and lacrosse go hand in hand in Canada, right? So, during the summers in Canada, they melt the ice, and so you play indoor box lacrosse. And so, like, everyone, we just transitioned from a hockey team to a lacrosse team. And so I grew up, started playing lacrosse when I was 10, and kind of just messed around and did that forever and still loved it. Not as much as hockey, obviously, but I knew the game well. And so my senior year of college, the club lacrosse team at Davis needed a coach. I had been helping out with them for a while and just kind of jumped into the coaching role. This is the women's club lacrosse team, I should mention. This is the women's club lacrosse team. And they needed a coach to help them get through their season. And so I took on that role with my now wife, which is kind of cool.
Congrats on that.
Yeah. So myself and Jacqueline coached this team, and we had a just great group of athletes. And we ended up, going all the way. We went in the 10 seed, came out the 1 seed, which was awesome.
Very cool.
So, U.S. Lacrosse National Champs, 2010.
That is cool.
It is cool. It's a lot of fun.
10 years ago, wow.
10 years ago. Makes me sound old.
How does, would you say that your athleticism or your propensity to do athletic things detracted from your ability to do grad school? Did you find yourself not having a lot of time, or would you say it was something that helped your work-life balance a lot?
The answer is complicatingly both. There was times that I feel like I should have not been doing so many sports, but I'm very happy with the way that my work-life balance went. I think the greatest thing about sports for grad students is the fact that when you are playing your sport, whether it's intramural, competitive, beer league, whatever it may be, it forces you to think about something other than science for an hour. So you can kind of clear your head. I see a lot of grad students who get into this detrimental spiral of like, they do their experiments, they go home, all they think about is their experiments, they come back the next day and they're just like stuck. And while this works for some people, I think it doesn't work for the majority of people. You need your alone time to kind of clear your head and refresh yourself. And so sports were that outlet for me. 4 days a week I'd play for an hour or two, and you're just forced to, like I said, think about sports, because when you're competing, if you're thinking about other things, like, it's not going to work.
I mean, it can totally be detrimental to your mental health, because, I mean, I've mentioned this on podcasts before, but science a lot of the time is just professional failure. Like, you fail constantly, the experiment doesn't work, or it doesn't go as you expected. And so if you're just constantly only thinking about, your failures or your experiment all the time, that can kind of not be so great, and certainly not great for the people around you. Your friends can kind of get sick of you sounding like a broken record, constantly talking about your experiment. So I agree, it's important to have some sort of outlet that can get you out of the science realm and get you into something else, not necessarily a healthier thought process, but just something to distract you for a little bit.
Yeah, absolutely. And yeah, professional failure is a great way to put it. I think it's really important for everyone out there to structure your thought process in a way that even though your experiments may fail, you have learned something in that failure. You've learned what didn't work. And if you can do that, then you're going to be a very successful grad student. You just take that failure, make it something positive, and transition that positive into your next experiment. But yeah, definitely, it's mentally draining to take months and have stuff not work every day.
Big time. Sometimes I'm like, why are they paying me to just fail over and over again?
I know, I ask myself that every day.
So kind of two things. What's next for the future of, this testing in your mind, or kind of like with this coronavirus testing?
Yeah, that's a very great question. Right now the university is kind of in limbo, right? We're starting to implement reopening procedures, but those have not been finalized by any means. The California State University system, the CSUs, I want to make that very clear, not the UCs. The CSUs have already said that they're canceling a majority of their fall classes and moving a lot of them online. I wouldn't be surprised if the UC system followed suit, but the key to reopening any major university system is can we test our students? Can we identify hotspots of infection? Can we come up with accurate transmission rate numbers? And so I think for university systems to really, really reopen is getting large-scale testing done in a very timely manner. So what does that mean for us? One, I think the biggest thing is increasing the capacity to test. UCSB has just shy of 30,000 students. You need to be able to do, I would say, accurately, those 30,000 students, I don't know, every 2 weeks, maybe? Every week if we could. But that means that you're looking at 3,000 plus tests a day, which is crazy.
Right.
That's just a lot of tests. The obvious answer to that is robotics. Can we automate that? Because automation works, 24 hours a day. You can just run these robots into the ground and they're not going to care. So I think if we can develop some sort of system or architecture that allows that to happen, then we can test massively and help the university develop procedures and processes to getting this open. I think it's doable. I think it's really doable. It's just a matter of working with an engineering team or a robotics company to make sure that happens.
Very cool, very promising. Not so much doom and gloom kind of sounds.
No,
like.
I think, I mean, realistically, like, I don't know. So our test, like we talked about Crests, we did a lot of development early, but it's actually being used to test UCSB students and faculty now. Like I mentioned, we're about 200 tests a day. We wanted to do a pilot experiment of 1,500 people just to see like what the feasibility scale is and things like that. So far, we've done it all by hand. And we're sitting, right around 900 tests right now in, I want to say it's been about 8 days worth of testing. Because we kind of worked in this ramp up, we did 30 one day, 50, 100, and so on and so forth. Anyways, in those 900 tests, so far, we've had no positives.
No way.
Yeah, which is great. That's great for us. It's kind of following suit with what UCSF has reported. They did a similar study. They did 1,800 tests in a community called Bolina. In 1,800 tests, they saw no positives.
Well,
so the university systems seem to be promising in their containment numbers, partly because their students, I'm guessing, and faculty have done pretty well isolating and wherever they chose to isolate. So, yeah, I'm hoping that we continue on this trend for our testing and, things stay flat.
Yeah.
Because that would help the UC system massively. I'm sure students want to get back to school.
Definitely.
Probably the first time that's ever been said, ever.
I know, what a weird dynamic. Like, yeah, students want to go to school, and I can't wait to get back in the lab. I want to go back to work so bad, but my lab's still closed, unfortunately, for the time being.
Yeah.
And then, kind of, what's next in the future personally for yourself? What do you foresee yourself doing?
Yeah, the big thing is kind of getting this postdoc going. At the end of my PhD, I was fortunate enough to work with Angela Patenis, who's a professor in materials, who kind of helped get me this joint postdoc between her and Max Wilson's lab, working more on like biocompatible materials, which is a field that I find extremely fascinating. So, unfortunately, I was only in lab for about a month when this whole shutdown got hit, and we've got some really cool ideas that I think would, kind of change the way that optogenetics works. So that's where I want to go, is like actually work on postdoc projects that I was hired to do.
Yeah.
Who knows when that's going to happen?
You mentioned optogenetics. You want to just talk about what that is briefly?
Yeah, so optogenetics is a way of controlling gene expression using light. So what people typically do is they engineer a protein that is derived from plants into the genome of a host cell. This way that protein then is light active. So when you shine light on it, it'll activate or deactivate a certain signaling pathway within the cell, which will allow you to either upregulate or downregulate certain genes. So you can truly investigate how a cell responds to a certain signaling pathway. It's kind of changed the way that we think about studying cell signaling for a couple of reasons. One, it gives you acute control over a single signaling pathway. Two, you don't have to knock out any genes. Like, gene knockouts are kind of been the classic canonical way of doing cell signaling pathways, but the problem is these signaling pathways are so intertwined that when you knock out a single gene, I personally believe that there's no way to be confident that the knockout is only affecting a single pathway.
Right.
People make that leap a lot in papers because there's no other way to think about it. But optogenetics offers you kind of a workaround for that. It's really fascinating. It's really cool.
I hope you get back to studying that sometime here shortly.
I do too. I mean, we'll see. I don't know. We'll see how long this persists.
But yeah, that about does it for us. I just want to say thank you again so much for coming on the show. Is there anything you want to say to the fans at all before we go?
Just stay safe. I don't know. I mean, I want this to end as quickly as possible, and people need to do their part to make sure that happens.
Well, again, thank you so much, Dr. Valois, for the insightful discussion. I just want to take this time to thank the listeners for tuning in and giving their support during the infancy of this podcast, especially as I try to get this thing off the ground. It really means a lot to have your support. So thank you so much, everybody. And, please reach out to me if you have any scientific topics you'd like me to discuss in the future. If you have somebody in mind who you'd like me to reach out and interview, I'm really open to any suggestions as I continue to roll out these episodes. And, once again, thank you so much. And until next time, this is Goggles Off.